Hello everyone. Today we will be learning about the science of hormones. So we'll be talking about how sex steroids work and some common applications of them in therapy. And the first thing that I want to do is remind you of something that I mentioned last time we were together, of chromosome puffs. So scientists eventually were able to isolate these steroid hormones and put them on various organisms to see what happened. And what scientists saw was that, for example, in fruit flies, when they treated them with hormones, they would see these chromosome puffs. So they noticed that hormones acted on the DNA and on specific segments of DNA that we call genes. And so, what scientists discovered is that these sex steroids have two primary functions. First, they work at the chromosome or DNA level, affecting gene expression. What do we mean gene expression? We mean what RNAs and protein. Here, what RNA and proteins are made. And this is happening kind of at a slow pace within cells. But these hormones also work on signaling pathways, so they help with communication in the cell. And this happens at a faster rate. Before we jump into this, folks, let's review some concepts we've already covered. So let's remind ourselves of the central dogma of molecular biology. So we've learned that DNA can make more DNA, and we call this replication. DNA is kind of the set of instructions in the cell, right? And DNA is converted into RNA through a process called transcription. And RNA we think of as the intermediate or messenger. Finally, RNA is turned into protein through a process called translation. And protein we think of as kind of the tool within cells. It gets stuff done. As another review, folks, let's remind ourselves of a couple things about DNA. Yes, it is the instructions for everything that happens inside of our cells. Individual instructions are called genes. This DNA exists everywhere in our body and in most cells inside of our body. And this DNA is how we pass down specific traits to our descendants. Now, structurally, DNA has a negatively charged backbone on the outside. On the inside, there are 4 bases that kind of match with each other, so that the A and the T go together, and the C and the G go together. And the order of bases is important. That is the instructions. But if we know the sequence of one strand, we will of course know the sequence of the other strand that it is connected to, because they complement each other. A always with T, and so forth. And then finally, folks, I want to remind us of some parts of a cell. We have the cell surrounded by the cell membrane. Inside of the cell is the cytosol. It's this kind of watery liquid that everything is in. And then, of course, the nucleus in the middle where the DNA resides. The 2 functions we'll talk about. Number 1, the work at the chromosome level is happening within the nucleus. The second pathway, this should be a number 2, is hormones work on signaling pathways, and so this can happen both at the cell membrane and within the cytosol. So we'll expect signaling to happen at the membrane and in the cytosol or cytoplasm. And we'll expect a gene regulation to happen in the nucleus. Next up, let's kind of walk ourselves through, more specifically, how these two primary functions work.